Today we'll talk about transmembrane prediction. Transmembrane helix. So essentially we have the issue that in a cell there's an outside, there's an inside, and we distinguish these two by cell membranes. Let me play at least this video here, if I can. Cannot. Yes, sir. Where's the no sound?
The cell membrane controls traffic into and out of the cell surroundings. All the cell membrane.
So this one here is about in the ballpark of 30 angstrom or 3 nanometer distance across one of these lipids. So that's an average lipid. So they do differ between different types of membrane. Prokaryotic, eukaryotic cells, and again, so this is the lipid. Here are the lipids shown with a head, and ultimately this hydrophobic reality changes the reality of a protein. While typically proteins sit in a solvent, and therefore at the surface of a protein you have hydrophilic residues. Now you change that into a situation where you have in the hydrophobic are on the inside. Now you change it to a situation where essentially you have in the environment of the membrane protein you have hydrophobic residues. That is the other part that I didn't I skipped, and here's a better way of looking at that from Arne Elafsson. The lipid is sort of indicated by this mesh here. That's the top end of the lipid, the lower end of the lipid by this end. The helices are going essentially perpendicularly through the lipid. So essentially this is a so-called 7TM, 7 transmembrane helices protein, so-called G-coupled receptor. And the reality in terms of energy, again, in a typical protein, there are hydrophilic residues on the outside, hydrophobic residues in, and here in the membrane protein essentially you have hydrophobic residues throughout. That's a very different energetic situation. Why do you need membrane proteins in the lipid?
Facilitate transport?
Yes, facilitate is a big word. I would in fact give it a bigger word to make it happen.
Well, there's active and passive transport.
Well, so again, the water gets through the membrane, but at a very low rate. But anything much larger than water doesn't get through, full stop. It's not that, in that sense, and in fact, let's go to the next two slides here. The first one to realize is if you have anything that is sort of inserted into the membrane, you can easily move it along the axis of the membrane. So in the direction of this arrow here, you can move the membrane helix easily through the membrane, right? You can hardly do it in this direction. So that's what the hammer here sort of is suggesting, that if you want to intrude into the membrane, that you cannot easily do. And that brings us back to facilitation of transport. Essentially, transport is not possible of larger molecules, certainly not on the level of DNA, protein, or whatever. Facilitate, ultimately you have the right word, but it sort of suggests that it would be possible otherwise. In fact is, but not really, okay? Yes, there's active and there's passive, but ultimately active and passive is all done by membrane proteins. Those are all gatekeepers. Think about those as gatekeepers. And in fact, there's even a mechanism, and this is work from Marek Basler in the Biocenter in Basel, looking at the so-called type 6 secretion system in gram-negative bacteria, which is built up of a lot of proteins, and that essentially is sort of, you can think about it as a pump or as an apparatus that builds up a lot of mechanical pump here. Then the pump suddenly collapses, and by this fast collapse, building up this ring here, this ring structure takes ballpark half an hour. It collapses within a fraction of a minute and produces a mechanical energy that pushes out something. It sits in the membrane and it pushes out. What would it push out, do you believe?
Hormones?
Yeah, this is you already. Hormone is a very specific answer, but proteins is sort of the much more generic answer. It's pushing out proteins. Why would a cell push out proteins? Or let's go one step back. I should have done that before here. I was induced by your question to go into the movement part. But the first question again. So we have proteins that facilitate transport. What kinds of transport is needed in a cell? What do you need to put into a cell? What do you need to get out? Yeah?
I guess signal proteins from other cells.
That is again, so this is, you're right, absolutely true. But let's look at the sort of first level survival scenario. Let's call it the pre-communication level. To live for a single cell, what do you need to do?
Nutrients?
Yes, so energy, whatever you call that, nutrients, energy gets in, and what gets out? Waste products. So essentially byproducts of many of the pathways of energy production, whatever it is, that one gets out. So you need, that is in fact why you need gatekeepers. You cannot have a cell surrounded by plastic. You need to have something that's totally open. And that gets to the next level. So these are all small molecules, right? So now why do we actually need proteins to be transported through this? And you had in two directions. So one direction is of course that something comes in. The something comes in typically can be disease, right? This is the way a protein from something else is a disease. But why are proteins secreted? Why do cells secrete proteins? Why do cells put proteins outside?
Yeah, maybe to communicate with their environment.
Exactly. And communicate with the environment has many levels. That has the level of sensing what is there, of going for food, getting the food. So there are various aspects of various reasons why you may want to communicate. Now, another way in which you could communicate, and that sort of is exactly what this one is about, is you want to. This is not like a communication, is it? Do you believe this is for communication? What do you believe is done here? Why is this energy built up to shoot something out? Who believes this is a sugar molecule to feed some other cell? Let's put it like that. Okay, what do you believe cells shoot out there? Do you believe this is the waste product shooted into the space?
Maybe some proteins to infect the specific cell.
Yeah, exactly. Yeah. This is exactly, in some sense, what Thomas said the other way around. So other organisms actually come in with their proteins, and this one here is sort of attacking other proteins. Exactly. It's shooting this into another cell. And this one, in fact, the reason why this machinery is so big is it spans, in fact, half the entire cell. This ring is so big that builds up, it fills the more than half the cell, and actually is so strong that it can shoot a protein through the membrane. So I said that you can hardly get into that membrane. This is a mechanism by which you can actually intrude through a membrane. So you approach the membrane and then you do the shooting mechanism. And that in fact leads to an attack. And now the green cell here is sensing the attack. It's not sensing the attack in the sense of it's dying and it realizes it's dying, but in fact it is immediately realizing where the attack is. And guess what is going to happen? It's going to assemble the same kind of mechanism and going to shoot back exactly at the same point. Right? This is really true. So Marek showed this. There are tons of movies on his website. It's a totally cool story. But now comes the coolest, what I found was the coolest part of the story. Now imagine that the green cell runs out of bullets. Right? So it has nothing to shoot back. What is it going to do?
To escape.
Yeah, very good. I do not know. Most likely this is in fact exactly the right answer. But the other surprising answer is it takes the arrows that were shot at it So it repurposes exactly what it has been shot with. So the orange shoots it with some arrows, and it sort of collects the arrows from orange and shoots back with the same arrows. So it uses essentially what it has been shot at with, the ammunition it has been shot with, hit with. Anyway, so this is an extreme example of how you can intrude into membranes. And bacteria really do that in the bacterial warfare. And so Marek Basler really has wonderful movies to unravel this whole story, an amazing story. When you look at 3D structures of membrane proteins, what you see is the assemblies of helices, groups of helices that are inserted into the membrane. And another example is here. In this particular example, and these two examples here, where what you see is essentially that these helices, this is two chains, is a homodimer. So these helices are sort of condensed inside of the membrane, and up here you see some sort of mushrooming structure, something much bigger. So it occupies less space in the membrane than up here. This is essentially the anchor, and there's much more to the anchor up there, and that much more to the anchor up there is the gatekeeper. You have examples, so this example here is the same here. You have examples where you have sort of helices that are just attached to the membrane, they're not entirely going through. And essentially all of these, there is another type of protein that goes through the membrane, the beta strand membrane proteins. We're not going to talk about it most likely due to time constraints. But this is the dominant type. Dominant type means most membrane proteins, the vast majority of membrane proteins in human are this type. Again, the other type is beta strand, all beta strands, and we have not observed any mix. So there's no single protein known where you have both helices and strands to go through a membrane. Now, when we do comparative modeling, I showed you that over the years comparative modeling gets better, or simply the coverage gets better because we have more and more structures known, right? This is the story here where just the blue part for which we can do comparative modeling over the years get larger. When we look at membrane proteins, you do not see a difference. And ultimately that reason is because this fraction here, the blue, is so small. It is so small, which is surprising given that in fact, when we look at drug targets, about 57% of all the drug targets are membrane proteins. So this is about 1,500 different drugs that are sort of known, and of these 1,500 drugs, 57% target the membrane proteins. Why would that be the case? Why would a drug target a membrane protein? Well, again, you see many examples. This is half full, half empty story, right? 43% are non-membrane proteins, so not all drugs target membrane proteins, clearly not. But why is it so predominant? What do you believe? Yeah?
They're the easiest to access.
They are the easiest to access.
Insert your drug into the cell, you can just have it connect.
So if you can do something before, so the first point of entry is the gate. And by the way, I should add this 13% to the 57 here, because essentially these are sort of proteins that are next to the membrane, right? All of this, the extracellular and the membrane proteins, they are the ones who sit somewhere easy to access. Just like Paul said, right? So if you want to control a cell, you try the first point of entry. Membrane proteins, secreted proteins, which essentially is the anchor part of the membrane protein just sitting above, mostly in this part, for these targets here. So this is exactly the point. It's the fastest way of entry. And typically, this is also the point in which, in fact, if a bacterium tries to get you, they get through the same anchor points. So if AIDS wants to attack, it does, or any kind of virus wants to attack, it will also attack first the anchor point, the receptor. And that means if you have the drug against the AIDS, you want to target the same receptor that AIDS is targeting, right? Which again is the entry point. So in some sense you're also mimicking, with this easy to access, you're mimicking the intruder. Okay, now This is another example of the GPCR, the 7 transmembrane proteins that I showed you before, which in fact, it alone is sort of 40% of all the drugs. Bless you, this is why this is such an important molecule. Here is a voltage-gated potassium channel. The voltage-gated potassium channel, so here you see an example where in fact it is sort of a loop. These are the membrane helices. It's sort of the loop here that holds, in this particular case, 4 potassiums in line. And then ultimately they are sort of pushed through one by one. The moment any molecule comes that is not a potassium, this will just not get through, right? It's very, very, very sensitive. The probability of getting through if you're not a potassium is much more than a factor of 10 less, a factor of 100. And it is formed by this beautiful structure here. Okay, now if we predict membrane structures, the first thing that we want to predict is really, where is the helix? Where is the helix and how are these helices oriented? And since it's a helix, the simplest way in which we can do that is we simply use the type of devices that we have talked about in the lecture so far. The methods that predict secondary structure for globular proteins. And if I applied that to a membrane protein, then this is what I get. So you have the observed membrane helix, and you predict something totally different. Why? Why do you believe this happens? So the simplest way is we look for hydrophobic residues, because we know that some residues are hydrophobic. There's a hydrophobicity scale, for instance the Eisenberg scale, that classifies according to more or less hydrophobicity. And we simply apply such a scale. But before we do that, let's just look at another scale. So that's the Eisenberg in green. There's a Kite-Doolittle, and there's a so-called GES score. You do see that these scores are not all the same. They differ, the bars differ. You also see that sometimes these bars differ substantially. So while in one class it's considered the methionine is considered very hydrophilic, and another one is more hydrophobic. So that is one issue. In fact, if we went to more than 3 scales, and now it's all scaled to 0 and 1, now I have 5 different scales, and you see the more scales you bring, the more diversity you get. In fact, there is a publication in '96 here where they collected, Tommy and Kanisha collected 402 indices, and they began to cluster them. And there is some large subset of all of these indices here that describe hydrophobicity. But in this cluster, you see there's a great variety. They are by no means all the same. So what seems to be a very simple solution, look at hydrophobicity, at the end of the day for you as a programmer, boils down to doing some Wikipedia search and pick that one. Unfortunately, there is no good way of doing it unless you took actually this paper and possibly took something like an outlier or a majority or some average. There are different ways in which you can do it, but averaging itself is also sort of changing the physics again. That may be the wrong way. Anyway, if you applied it to this particular example, you see that in fact the hydrophobic, and that is, I assume this is the Eisenberg scale here, that essentially the helix, the transmembrane helix here, is sort of found by the hydrophobicity scale. So this solution is actually working. Gunnar von Heine sort of refined this a little bit, and he said, okay, the hydrophobicity is something that is a scale here. Here is scaled from -4 to +3. This is the protein position, so it's a protein from 0 to 400 residues long. And now you could, the simplest way to design transmembrane helices would be to say, okay, there's a threshold. If it's above the threshold, say 1, I call it transmembrane helix. And that would give you these transmembrane helices up there. But Gunnar sort of introduced a second threshold and found a way to sort of optimize the choice of these two thresholds by some additional information. And that additional information essentially is the positive inside rule, and the positive inside rule is the following. So he's going to look, he looked at the composition of all the regions next to the membrane inside, and the composition of all the regions next to the membrane outside. And he asked, are there different residues used? And what he published a while ago, in '86, is that on the inside you have more positively charged residues. So the red is more positively charged than the blue. Okay? And that ultimately was, it is really only the positive charge, the R and K, residue R and K, that differ. Nothing else. It's the D and Q, so there's no balance of charges. I sort of repeated this on a much larger data set about a decade after this, and the only signal that existed is really the one he found. Now that means that essentially you can sort of optimize which threshold you use such that you make the positive inside rule maximal. So you lead to a situation in which the helices are assigned such that the positive charges, or the difference between the positive charge on the inside and the outside, is maximal. Very simple algorithm, right? And that allows you to sort of refine a different threshold. So you have two thresholds for hydrophobicity and do the same thing. And that has been for a long time one of the most successful methods to predict membrane helices. Or you could do the same thing that we did before, this using evolutionary information as input from alignments, as input to some neural network, but this time train it on transmembrane proteins. So do everything the same way we did before, except for now you have two output units, transmembrane, not transmembrane. Two output units shown here, transmembrane, not transmembrane. What I show again here is the sequence, so residue 0 to 400. No, it's 300 here, 100, 200, 300. And this is essentially the output from this unit. Now, you remember there were two layers of neural networks. One did sort of sequence to secondary structure, and one did secondary structure predicted to secondary structure again. The first level had two short helices predicted. It did not see that helices have to have a minimal length. That's why we introduced the second level, and the second level sort of saw the correlations. Okay, you remember that story? Good. Now, the problem is the second level works too well. So what you see here, if you just look at something that is above the threshold of 50%, you see that you get to a situation from this point to that point here that is much more than 25 residues. So now you create, so the simplest way to translate this to transmembrane helices is you define another threshold above 50, or whenever this one is higher than this, which essentially is above 50, because I believe they have been strictly normalized in this particular case. Anyway, so then that would give you a membrane helix spanning from here to here, and given that 2 points here are 50 residues, this membrane helix would be more than 50 residues long. So twice as long as it is supposed to be. And how can you... So essentially what it means is that you do the first level, they're too short. You do the second level, they're too long. What can you do? So what I did try is a greedy approach. And this image here is a much simplified version of the one before. So in this much simplified version of before, we begin by showing all possible segments between 17 and 25 residues. I do not know how many that would be in this particular case, but certainly in the ballpark of 1,000 or more. Thousands maybe. So I pretend that this is 8, not thousands, but 8. Those are all possible transmembrane segments that have an average above 0.5. Okay, this is the way I'm going to explain my method to you. In a normal case, you will not find that. Completely made up example. But it's much easier to show. Okay, we have 8 segments. The number that I show here is the average of the t-score for these, per residue t-score for these 8. Okay? And again, these 8 differ in length. Essentially, they are all the ones 17 to 25 that fit into the landscape. Now, if I assume that my protein has 1 transmembrane helix, which one would you pick out of this pool of possible helices?
Where are the 8? I only see 6.
1, 2, 3, 4, 5, 6, 7, 8.
Oh, well, what's that one down?
Let's just go
okay.
We begin there.
The one with the highest.
Okay, I should have an animation. Okay, I learned today that there are many things wrong in the way I present that slide.
It looks like beautiful helices, which is why I was counting the candy canes.
This is sort of simple hacked refinement. So this is the pool of all possible 8. And again, the protein runs N-terminus to C-terminus. We have 1, 2, 3, 4, 5, 6, 7, 8 possible transmembrane segments. The number in this segment indicates the average over this T curve. Okay? Now, if you predicted that the protein has 1 transmembrane helix, which one is it? Which one would you pick? That's very obvious, right?
The score?
Yeah, sure. You'd pick the best. You pick the one with the highest score. So, this one, right? If you predicted that your protein has 2 transmembrane helices, now you have an issue. So let's just keep it as the simplest, greedy, dumbest algorithm that you can. And it's not dumbest, but it's a greedy algorithm. So in a greedy algorithm, you will argue, okay, the second largest one is this one. I cannot pick that because that overlaps. It's impossible, right? So, and I will not shift this around because that is why it's greedy. I will keep whatever is best. If it's 1, it's that one. So if I predict 2, then I have a good reason to argue. If I go through to a greedy solution, it's a good argument to say, well, since that is the best for predicting 1, it will also be the best as the first of predicting 2. In a greedy way, it's right.
Wouldn't you just want to maximize the score?
The
overall score?
But then the story gets slightly more complicated. Then you have to essentially put in hidden, exactly. And then boom, with the word all, you got yourself into exactly where the problem is. All is always high. And this one here is extremely fast. And you then take simply the second best, which is this one. And then if you believe it's 3, then pick the third best. And ultimately the algorithm converges exactly at the point when there is no fourth best. So in this particular case here, there's none that is not overlapping to something that we already picked. So we cannot take more than 3. And all we need to see is that the probability for 3 is higher than the probability for 2. And if that is the case, the probability through the landscape. Because once we have the model, we can easily compute the probability for the entire thing, right? And then I can simply say, okay, if that is higher here, I predict the protein has 3 transmembrane helices, and they are here. Okay? Now there's also the positive inside rule that we can apply to see how it's oriented. And for the orientation, I will simply count the positive residues on that side, sum those, positive residues on this side and that side here, sum those, and that will simply give me the orientation. Whatever number is higher, the odd loops or the even loops, I will predict to be inside. Is the algorithm clear? There are a couple of implications here. One is that the way I use as a criterion here for best, I use the average. This will of course sort of favor short transmembrane helices. And that is suggested by the fact that many transmembrane helices are pretty straight. So if you can get 17 residues to go through the membrane, you will use 17 and not more. So with more tilt, this is sort of changing, but the argument is if you do really have a tilted helices, then, and you do in fact have 19 that fit, then most likely the signal for the 19 will be stronger. Because evolution will favor that there will be a very strong signal. If evolution has sort of optimized this structure, then it will favor to change the sequences such that in fact they will get higher signals, and that in fact is the case. But that still is one issue. So one issue is that I favor shorter segments, and the other one, of course, I don't find the best model, the best possible model for the entire path. I'm not computing the entire path for all possible best models. I'm just giving one greedy solution. And that greedy solution for this particular case here worked very well. And ultimately, sort of the idea to go into more than one greedy solution is this TMHMM that came again from Gunnar von Heinje, Eriksson, and Anna Schroch. So Anna Schroch was the one who, now at Copenhagen University, brought essentially hidden Markov models into biology as alignment tools initially, and here applied it to membrane proteins. And the way it's done essentially is you simply have different states, and in these different states you model the part that is the membrane, the helix core, you have capping of the helix, you have cytoplasmic loops, then you have this part that is really the globular protein, and you have a similar story on the non-cytoplasmic side. So you have different states that you can model by different background probabilities and sort of really learn a detailed hidden Markov model through this. And the hidden Markov model works very well. There is a slightly different approach here from Gabor Tushnadi and Istvan Shimon. HMM top, and the difference really again is using HMM. The difference really is in the way the detailed modeling of these loops is done.